Good morning, once again, everyone. Hello, I am clinical pharmacologist Dr. Arun. Through my channel, Clinical Pharmacology CPT Pandora, I am introducing the different innovative topics on this lectures. Out of which pharmacogenomics, I have already given introduction as a pharmacogenomics uh, section and pharmacogenomic methods. In these two oncoming lectures, I will review the important points of the pharmacogenomic and precision therapy for all of you. So, any doubt, you can put comments on the chat box. I will review the comments and will give the explanation for each. So, now we can start the topic in a brief way. So, all of you know I am a clinical pharmacologist. Got my DM from AIMS Jodhpur. This is one of my DM presentation topics, and also I have reviewed this topic for many of the audience by the time. So the main abstract of this topics are pharmacogenomic concept. I will review the what is pharmacogenomics. Actually, what is the meaning of pharmacogenomics? Then pharmacogenomics, what is the relation of pharmacogenomics with the pharmacokinetic, drug transporters, receptors, and pharmacodynamics? Then pharmacogenomics can foresee the adverse drug reaction or not. Then personalization of therapy in different domains, in oncology, in antibiotics, in immunotherapy, and in psychiatry, in cardiology, in neurology, all the departments, whether the pharmacogenomics has a role in different disciplines. And pharmacogenomics as a translational platform for the drug development. And pharmacogenomic, what are the clinical research and study designs of the pharmacogenomics? And genomics in the pharmacogenomics, what is the methodology? Pharmacogenomics, whether it is a future medicine and hope of precision therapy. So all this I will be review in detail. So by the section, I will be each slide I will be reviewing each concepts. So we move on to the next. So these are the different developmental phases of the science pharmacogenomics. Before 2006, Herceptin receptor approved with the companion of DNA diagnostic. Several FDA approved pharmacogenomics techniques were there. Large industry invest and regulatory interests are there. Industry FDA academic consortia being created. And by the oncoming years in 2008 and 2010 and towards the 2012, slow limited pipeline growth, like significant increase in pharmacogenomic activity to provide the competitive advantages. And further integration of stratified medicine into developmental programs, more government collaboration, more external incentives, and recent higher pressure, all are there. And towards the 2016 and 2018 and towards the 2020, robust pipeline is there. Pharmacogenomics driven disease stratification, treatment, prognostic implication, and diagnostic implication, all there in the towards the late 2020s. It is in the COVID pandemic era. It become more robust. So, what is genomics and what's the relation with pharmacogenomics? So, the human genome consisting of 3 billion, a total of base pairs, is 3000 MB total length. And what is a gene? So, these are the basic concepts you have to review before getting into the concept of the pharmacogenomics. So, gene means a gene is a sequence of DNA or RNA that encodes for a molecule that has a function. During gene expression, the DNA is first copied into RNA. What is allele? One of the two or more alternative forms of a gene that arise by mutation and are found at the same genetic locale. What is SNP? So SNP is a single nucleotide genetic polymorphism. It is more than 1% population with inherited a nucleotide that differ from the most common sequence wild type at the position in the genome. And then what is the database of the short genomic variations? That's also same in the 50 base pairs repositories. So what is SNP? SNP then that cause amino acid change are referred to as non-synonymous SNP, NSSNP. Then now we are reviewing about Hardy-Weinberg equilibrium and linkage disequilibrium and its relevance in the population genetics. So what is haplotype? So haplotype is a combination of S and P that are commonly inherited together in the same region of DNA. So linkage disequilibrium analysis for analyzing haplotype. We are using linkage disequilibrium analysis. The HapMap project has revealed a block-like structure of linkage disequilibrium, well as the existence of area of low or high recombination rate. And this has helped to identify so-called tagging or tag SNPs. So what is mutation? A mutation is change that occur in our DNA sequence either due to mistakes when DNA is copied. The Human Genome Project and the Descendant, the International Map Project, in 1000 Genome Project and ENCODE Project traces many types of variation in the genome. So International Map Project, 1000 Genome Project, Exome Sequencing Project, along with the publicly available database like PubMed, Ensemble, University of California, Santa Cruz Genomic Browser, and the National Human Genome and Research Institute become pillar stone for genetic data mining. So this all things we have reviewed, what is mutation, type point mutation, out of which silent mutation, missense mutation, nonsense mutations are there, insertion deletion is there, large scale mutation like amplification, deletion, translocation, inversion, loss of heterogeneity is there. So what is UCSC browser, genetic browser gene mapping? So this is a snapshot of the UCSC browser. You can see the chromosome 15, different nucleotide base pairs, 50 base pair region is highlighted this area, reference sequence is highlighted in this area, and this is SLC gene, SLC228A1 gene. It's got different variation between human, rhesus, mouse, dog, elephant, chicken, zebrafish, all are depicted in this diagram. This is one snapshot of this browser. So various genetic sources, we have already discussed it. We can get the genetic source from National Human Genome Research Institute, GeneBank 100 Genome Project, DBSN, DB variant, clinical variant, database of genomic variant, encyclopedia of DNA elements, roadmap, epigenomic project, pediatric cancer project, pharmacogenomic knowledge base, clinical pharmacogenomic implementation consortium, US Food and Drug Administration pharmacogenomic biomarkers, cytochrome P450 homepage, and human CAP allelic non-nomenclature committee, UCSF FDA transporter. These are the sites we can get the genetic database. So now we review the different aspects of the pharmacogenomics and what are the relation of it with the pharmacokinetic, dynamic, and the transport proteins. So all that thing. Out of which all of you know about the metabolism of the drug. Cytochrome P450 enzyme system is a major pillar stone of the metabolism of the all the drugs and synbiotics that take place in the liver. There is a huge number of inter-individual variation in the drug action due to this genetic polymorphism of the CYP system. So SNP single nucleotide genetic polymorphism CAP2D6, 2C19, 2C9, 23AB, and its inter-individual variations in drug metabolism are there. There are fast and slow acetylators. For example, the epic examples are warfarin, CAP2C9, and VKOR-C1 gene SNPs. Douglas CAP2C9 SNPs. Irinotecan UGT1A1 activity. Omeprazole CAP2C19 polymorphism. Hexamethonium plasma cholinesterase deficiency is there. It is also genetically related. Azathioprine mercaptopurine is intimately depend upon the thiopurine methyl transferase metabolism gene, TPMT gene, genetic polymorphism. So these are some of the list of common mutation and functional effects on CAP metabolic enzyme. CAP2D6 VA 4 metabolizer, CAP2D6 4B 4 metabolizer, CAP2D6 5 4 metabolizer, CAP2D6 1 XN ultra rapid metabolizer, CAP2C19 2B 4 metabolizer, all 3 are 4 metabolizer, and so on. You can see ultra rapid metabolizer and 4 metabolizer listed. And this is another one. DNA-based biomarker of enzyme activity considered as valid biomarker. So CAP2C9 is warfarin. Drug metabolism is depend upon the CAP2C9. So if you are giving a maintenance dose, a time to reach outcome measures, we can measure the maintenance dose, time to reach a stable dosing. Study results indicate that patient with this 2 and 3 variants maintained the lower doses and took longer time to reach the stable dosing. Another example, CAP2C9 proton pump inhibitor is metabolized by CAP2C9. So plasma levels higher in poor metabolizer. Gastric pH, gastroesophageal reflux disease cure rate, higher dose 40 milligram showed no difference. And CAP2D6 could be morphine formation, analgesics effect of the codeine higher in EM variants. UGT1 to A1 help in the metabolism of the aeronautic, and grade 3 bar 4 neutropenia is more in UGTA1 7 bar 7 or 6 bar 7, more frequent than in 6 bar 6 variant. Pharmacokinetic parameters AUC ratio of SN38G or SN38 is with the UGT1A1 28 and 6 variant with reduced ratio. Then TPMT is the important gene and enzyme involved in the metabolism of 6-mercaptopurine. So the dose limiting hematopoietic toxicity is more in with the TPMD polymorph variants, glycozygotes. So I think this section I will stop by the next slide, and I will continue in the next second part of this lecture series, the other aspects of the pharmacogenomics. So by this slide, we can conclude this discussion. So pharmacogenomics and drug transporter mechanism. Gene products that are the drug receptors and target have an important role in pharmacogenetics. So multi-drug resistant protein receptors, gene mutations are there. Solute-like protein superfamily mutations are there. Serotonin receptor polymorphism predict the drug responsiveness to as well as over risk of depression. Beta receptor polymorphism linked to asthma responsiveness. Polymorphism in HMG-CoA reductase linked to the degree of lipid lowering effect by statin. And ion channel polymorphism have been linked to the risk of cardiac arrhythmia like long QT syndrome, LQT1, LQT2, LQT3, Brugada syndrome, short QT syndrome, and drug-induced toxicity points. AC enzyme polymorphism can predict the renal function responsiveness to following ACE inhibitors. So by this, I am concluding this discussion. The next section, we will continue with the slide. Thank you. So keep on watching the next series of lectures. We will review in depth of the pharmacogenomics and the concept and ideology. So once again, thank you. Okay.